Hello everyone. Today we are going to be doing the extraction and the processing of DNA by PCR from microorganisms. This is for the MDLT 499 course, and I am your instructor, Renan Thompson. The first step that we use in the analysis of DNA is the extraction of DNA from our cells of choice. Today, the cells of choice that we would be using would be gram-negative bacterial cells. The basis for the extraction of DNA is the removal of any item from the cell that is not DNA. That is, we would be removing proteins, we would be removing lipids, and we would be removing RNA from the cell until we are only left with DNA. Before we do that, we must lyse or break open our cells to be able to expose all of the internal cellular components. Only after all of these steps have been completed would we then be able to analyze our DNA by whichever polymerase chain reaction method we choose. So today, our materials and reagents would include, but are not limited to, our pipettes and pipette tips of various sizes, of course our cells of choice, our gram-negative bacteria. We would be also working with tris acetate EDTA buffer, which is ideal for working with DNA extractions. We would be working with lysozyme. We would be working with proteinase K. We would be working with 2% Triton. And our materials would include our pipettes, pipette tips, petri dishes, beakers, Eppendorf tubes, flasks, conical flasks, PCR tubes, and test tube racks, as well as our centrifuges and our vortexes. In order to begin our DNA extraction, we would of course start with our cells of choice. Today, our cells of choice would include gram-negative bacterial cells. These cells have been grown overnight, and a few loopfuls have been suspended in tris acetate EDTA buffer. Tris acetate EDTA buffer is ideal for DNA extraction, as previously mentioned, because it preserves the pH and isotonic balance necessary for the preservation of DNA. It also prevents the action of DNases, which are enzymes that will degrade DNA. So, we would have vortexed our sample until all of our cells would have become suspended in 1 milliliter of our TAE buffer. In order for us to continue with our protocol, we would then take 135 microliters of this suspension and place it into a fresh Eppendorf tube. We would then take out 135 microliters of 2% Triton in TAE buffer. The Triton acts as a surfactant that would break open the protein cells and release the cellular contents. We would then mix by vortexing. After we have added our surfactant, we would then add 30 microliters of lysozyme to our mixture. What this does is that it assists in the lysing of the bacterial cell wall. We would then incubate this mixture, vortex, and then incubate this mixture at 37 degrees Celsius for 30 minutes. The reason why this is done is because lysozyme, being an enzyme, has an optimal temperature of 37 degrees Celsius in which to work. our water bath has been preset to 37 degrees Celsius to allow for this process. After we have taken out our mixture from the water bath at 37 degrees Celsius for 30 minutes, we add 15 microliters of our proteinase K and incubate vortex and incubate for 2 hours at 65 degrees Celsius. The reason why we add proteinase K is to aid in further digestion of the cell wall. We do this at 65 degrees Celsius because 65 degrees Celsius is the optimum temperature for the operation of this particular enzyme. We will now incubate at 65 degrees Celsius for 2 hours. After we have finished incubating our mixture with proteinase K at 65 degrees Celsius for 2 hours to be able to complete the full digestion of the cell wall and everything that is not DNA, we would then spin down our mixture with a balance in our centrifuge for 30 seconds to allow any pellet to centrifuge to the bottom. We would then remove the supernatant, which will then be used as our DNA and can be stored at minus 20 degrees Celsius for 3 months or at minus 80 degrees Celsius for up to 1 year.